This is a production of Cornell University.
Okay, my name's Seren. I'm going to talk about preliminary results today for a multivariate genomic-wide association study for carotenoids and dry matter, which are two negatively correlated traits in cassava. So you probably know that cassava is a really important food security crop. It feeds over 800 million people globally. It's a really important source of calories, mostly through carbohydrates, but it's pretty low in micronutrients. And so populations that rely heavily on cassava-based diets often have high rates of vitamin deficiencies, including vitamin A deficiency, which affects 1 in 4 children in sub-Saharan Africa. There are some yellow cassava varieties that accumulate carotenoids, which are some of which are cleaved in the human body to form vitamin A. So they're called provitamin A carotenoids. And what's great about yellow cassava is that it can provide a sustainable source of provitamin A for people in sub-Saharan Africa. And because of this, breeders have been investing over the last 20 years in breeding improved varieties of yellow cassava that can provide provitamin A and meet other agronomic trait needs. Unfortunately, there's a trade-off between carotenoids and dry matter content. Dry matter content, I'll abbreviate DM throughout this presentation. It's a really important driver of consumer acceptance. It influences the dry yield for flour and also the textural properties of cassava. And it's one of the key traits to target for improving for yellow cassava varieties. So this negative correlation, it's been persistent in the African breeding programs, and that's been a problem for producing high dry matter varieties that also have high carotenoids. Previous GWAS studies have identified that there's a locus on chromosome 1, which is associated with both carotenoids measured here by chromometer and also dry matter content. Because there is a strong candidate gene at this locus for both carotenoids, which is phytoene synthase 2, the key rate-limiting enzyme in carotenoid synthesis, and also sucrose synthase, a separate candidate gene that's likely to influence dry matter content. It's thought that this locus represents a genetic linkage between these two genes, and that's driving the negative correlation between the traits. There's also been molecular studies looking at the transgenic expression of PSY2 that suggests it has a pleiotropic effect on both carotenoids and dry matter. So there's a bit of conflicting evidence in cassava for whether linkage or pleiotropy is driving this relationship. And the objective of most of my research is trying to understand the genetic relationship between carotenoids and dry matter and the implications for developing breeding strategies that can help us increase both carotenoids and dry matter. So there's a few different hypotheses about how these traits are related both at the chromosome 1 locus or genome-wide. So first, for linkage, it's the idea that there are two separate loci that are closely spaced together that separately affect carotenoids and dry matter. There could also be horizontal pleiotropy or biological pleiotropy, where a single locus affects both carotenoids and dry matter directly or mediated. And then there could be vertical pleiotropy, where a single locus affects carotenoids, which then, because of physiological connections, affects dry matter, or vice versa. And so testing which of these relationships fits at chromosome 1 is important. And if we think that it might be vertical pleiotropy, which seems more likely than horizontal pleiotropy in this case, we'd also expect that other loci that affect carotenoids genome-wide would have a similar association with dry matter because of if they do have an inherent trait relationship. So for this study, we're doing a multivariate genome-wide association study to look at loci genome-wide. Using a multivariate analysis, we can increase statistical power by taking advantage of the correlation between traits. And we can also test hypotheses about the distribution of jointly associated loci across the genome. So we want to know if there are multiple loci that are jointly associated with both traits besides just the chromosome 1 locus that we already know about. And if so, whether those jointly associated loci affect the traits in opposite directions more often than expected by chance, which would suggest that there is an inherent relationship between the two traits. So for this study, we genotyped 378 accessions sampled from the yellow cassava breeding population at the International Institute of Tropical Agriculture in Ibadan, Nigeria. And Dr. Elizabeth Parks is the breeder who has been working in that population. And they have, together with her team there, they have collected child data over the last several years, which include carotenoids measured by spectrometer, which is a quantitative method that's improved from previous color chart methods that are more qualitative, and also dry matter content. And with this data, we are using two methods. The first is a multivariate linear model, which is more of a traditional GWAS. And the second is a Bayesian model comparison, which is going to help us interpret the results of these associations. So for the first method, for the multivariate linear mixed models, we're doing a two-step approach. First, we calculate BLUPs, and then we test associations between SNPs and BLUPs, accounting for relatedness, population structure, and the major effect locus on chromosome 1 that we already know about. In terms of the linear model, the first step looks like this, where we account for trial effects and calculate BLUPs using Box-Cox transformed phenotypes so that they're normalized. And then we take those BLUPs and put them in an association model where we have, in addition to the effect of the SNP, we're also accounting for genome-wide relationships, also the relationships of the haplotypes at the PSY2 locus and the haplotypes at a region in chromosome 1 where there are integration diagnostic markers or IDMs. And those are tagging a haplotype that causes a lot of population structure on chromosome 1 and is also known to affect dry matter. So we account for those effects so that we can try and reduce confounding from population structure and also identify loci other than the major effect that we already know about. So for this Manhattan plot, there's the negative log 10 p-values from those associations on the y-axis. The cutoff here, the dotted line, represents a modified Bonferroni cutoff, which is calculated with alpha of 0.05 and the estimated number of effective SNPs. And you can see that there are multiple peaks above this significance threshold, not just on chromosome 1, which suggests there are joint associations in other parts of the genome as well. What's challenging about this more traditional multivariate GWAS approach is that when you have a single p-value for these associations, it's difficult to interpret whether that SNP is associated because it affects both traits jointly, or if it really just affects one trait. These associations can represent strong associations with carotenoids and or dry matter. So even if it's a very strong association with one trait, it might get picked up here as well. So to help us interpret this, we're using a method by Matthew Stevens called multivariate Bayesian imputation-based association mapping. And the idea behind this approach is to compare models that bin the two phenotypes into these three possible partitions that are directly associated with the SNP, indirectly associated with the SNP via correlation with the other trait, or unassociated with the SNP. And so it calculates probability for each of these possible partitions and a Bayes factor, which represents the support for a particular partition compared with a null, which would be that neither trait is associated with a given SNP. So then how this looks in practice, because the Stevens algorithm doesn't allow you to fit covariates, we have to do it in two steps again. So the first step is similar to the previous model I showed, where we are accounting for the effect of the PSY2 haplotype, the chromosome 1 integration, and also the rest of the genome relationships using a leave one chromosome out approach, where we're taking the residuals from this model for which one chromosome has been left out, and then fitting that with the SNPs on that chromosome, which were left out. And so for each SNP, we can then see which partition has the highest base factor, representing which model fits the data the best in this case. So in this figure, it's the same Manhattan plot I showed earlier from the multivariate linear mixed models. And the color that's overlaid on top is representing the top base factor that came out of the BIM-BAM approach. So in red are SNPs, which their highest base factor was corresponding to the direct partition, which is that the SNP is directly associated with both carotenoids and dry matter. There's also a peak that's in green, which represents the partition where the SNP is directly associated with carotenoids and indirectly associated with dry matter. So once you account for the effect of carotenoids, there's no residual effect for dry matter. We can also look at the effect direction of these SNPs. So in red are SNPs that their effect estimate for carotenoids and dry matter are in opposite directions, and the blue color is for the same direction. So you can see that all of the SNPs which fall above the significance threshold are in red, which means that they affect the traits in opposite directions. So in conclusion, it looks like we have significantly associated loci that affect both carotenoids and dry matter in opposite directions that are across the genome, not just on chromosome 1, which suggests that there's a genome-wide negative relationship. The direct model fits best for most of the jointly associated loci, except for the one on chromosome 10. And also there's still a peak on chromosome 1, even after accounting for the PSY2 haplotype. So this suggests that there might be multiple causal loci in that region on chromosome 1. And our next steps are to evaluate candidate genes and follow up on some of these pleiotropy hypotheses using other methods like a network analysis that we're doing. I'd like to thank my committee and the IATA Cassava breeding unit team who collected this field data over the years, led by Dr. Parks and Dr. Gabby Mbanjo. And those are a picture of some of my lab mates at the Next Gen Cassava conference last year. Thank you. Any questions? Ed?
For the really nice work. I was wondering, is it possible to get access to data on ABA if it's kind of like a pan carotenoid thing, showing how much gets into ABA, that would, kind of, I think, kind of explain the million kinds of results.
Yeah, Ed was asking if it's possible to get ABA abscisic acid data, which is downstream of beta carotene and is one of the possible mechanisms for which carotenoid pathway might affect the dry matter and starch pathways. We don't have large data sets on that, but the data that there are for where abscisic acid is measured shows that there's a positive association between total carotenoid content and abscisic acid and with lower dry matter. So the correlations support that idea, but it's very hard to, I think, test abscisic acid in a high throughput manner, although Tim Sutter may have different ideas. So the starch, there's 2 starch biosynthesis genes near PSY2 on chromosome 1 that have been hypothesized as candidate genes. The first one is sucrosynthase, which is bidirectional in converting sucrose into starch. There's also another enzyme called UDP glucose pyrophosphorylase that's involved in the starch pathway, and those are both really close to the vitamin synthase 2 gene. So either of them are likely candidate genes to affect starch synthesis, but there have been no variants tested in those genes that are potentially causal.
Great, thanks for your answer, Seren.
Any other questions? Yes.
Just want to clarify the definition for the vertical polytrophy. Is that means it's sequential?
Yeah, so the question was clarifying vertical pleiotropy. So sorry. So for horizontal pleiotropy or biological pleiotropy, the idea is that a variant like phytoene synthase 2 has a direct effect on dry matter, which in this case is kind of hard to imagine, but there's other cases in which it might be possible to find a gene like a transcription factor that has direct effects on multiple separate traits. For vertical pleiotropy, the idea is that there's a single variant, for example, in PSY2 that affects carotenoids, which we know it does. And then because carotenoids might affect dry matter, for example, through the abscisic acid connection that Ed mentioned, then that variant would also affect dry matter, but only via its effect on carotenoids. And so once you account for the effect of the SNP on carotenoids, there's no more effect on dry matter separate from that. Does that make sense?
Maybe simplify to say horizontal is more like parallel and the vertical is more like a sequential.
Yes. Yeah. Horizontal is like parallel, vertical is like sequential. Yeah. Okay. Thank you. We can pass it to the next speaker.
Alrighty. Many of you already know that much of my research has to do with gourds. And today I'm going to specifically focus on bottle gourds. As part of my research, I'm characterizing the USDA Ligenaria collection. Okay. So first I want to talk about the area I'm focusing on. So most of my work is dealing with Southeast Asia. And there's a lot of discussion on how to define Southeast Asia as a region by scholars, whether it really should be its own region, whether we should think about it kind of between regions as a connector. But for my purposes, we're going to talk about Southeast Asia in these 11 countries, mostly focusing on mainland Southeast Asia, so Cambodia, Vietnam, Thailand, Myanmar, and Laos, but a little bit in the Philippines, and also in Indonesia. So why bottle gourds? So to me, bottle gourds are really interesting because it's believed to be one of the first domesticated crops in the entire world. And I believe part of this is due to its duality. So bottle gourds are quite unique because you can eat the leaves, you can eat the flowers, you can eat the fruit, but when the fruit matures and the plant senesces, it has a very thick rind. And this rind can be used for many different things. Now, traditionally, if you might have some familiar with bottle gourds, the most common thing to think about is its use for holding water. So we might think of a bottle gourd something like this, where we might cut a hole in the spout, we might cut a hole in the side to allow air to come through, and we would use this as a drinking vessel. So I have some show and tell, so I'm going to pass some things around for you guys to see. But when we look at the history of bottle gourds in Southeast Asia, it's quite diverse, the different uses. I'm going to keep doing that. Oh, my video's not going to play. Okay. So one of the uses you see across especially mainland Southeast Asia is its use of musical instruments. So in most countries, you can find these instruments by different names. So we have classifications such as the zither. So the idea is you have this long stick that has strings attached. It can be maybe one string, it could be up to like 6 to 8 strings, depending on the country of origin. But the really important thing in most gourd instruments is that it acts as a resonator. And a lot of instruments actually come from gourds originally. So things like banjos, we have these Vietnamese lutes played by these Thai women, where the base of the instrument is the gourd. And it also can be used kind of as an amplifier with the human body. So in the case of the zither here, we're going to use our imaginations. But if this is my gourd resonator, I can hold this to my chest. It's going to be cut in half. And as I play it, this will act as a resonator. And then also my chest is a resonator to increase the volume. So before we have electronics, you know, in the pre-modern era, or playing musical instruments, gourds are our natural amplifiers, so we can play to a large audience and everyone can hear us. A second use that you might think of gourds being used as is as garments. So two of the more common examples is in the Philippines, these Tao Bengal hats, and also in West Papua as kotika. So this would be a common gourd you might use for these traditional Filipino hats. Typically, again, you cut these in half, you'd braid up some reed to act as a soft interior. And depending on where you're gonna wear it, you might just keep it plain and use some sort of resin to preserve it and wear out in the field. Or if you're going to wear this for a special occasion, you might carve into it, create designs, you might paint it. And unfortunately, a lot of times due to imperial powers in a lot of these Southeast Asian countries we colonized, many of these practices are dying out. So this Filipino man, Teofilo Garcia, he is one of the last people actually making these hats in all of the Philippines. Same thing with the use of the kotika in West Papua. It's very important culturally. This is like the traditional garment that men will wear. You might use something like this to make your kotita. Depending on the shape you want, which partially does correlates to tribal identity, you may keep it curled. You probably don't want it this curled. But typically you might have something, you might wrap this around a log. If you want it straight, you would tie a small rock to the end so it grows straight. Then once you get your desired shape, you're going to harvest your gourd, put it on coals, gut it out, and then you let it smoke in the roof of the men's house until it's preserved. After that, you might decorate it. So here are these Kanak warriors. I believe this one has a like braided rattan covering. This one's a little bit more simple in design. People might put furs on it. They might paint them. And part of it is for use, whether you're at a festival versus working out in the field. And part of it is also the tribal identity, as I said before. So another place we see gourds is as amulets and as relics. And this photo here is one I actually took in Thailand when I was visiting a farm that grows bala gourds, which there they call namtao. And they're seen as a sort of protector. So at each corner of their house, they actually had one of these gourds hanging. And it's supposed to kind of ward out evil into their farm and keep their plants growing well. And although this is not part of Southeast Asia, there are also a lot of Chinese communities that over the centuries have come to live in Southeast Asia. And in many of the Chinatowns and other communities, you can find these, especially near New Year's, where in China, from what I was told by the ladies, is typically bala gourds are meant to bring good fortune to you. So you might hang a bala gourd in your house or in a car or to bring your family more money for the new year. Another way that bala gourds are in Southeast Asia, sort of this religious folklore area, is in these people of the gourd stories. So there's this idea that mostly mainland Southeast Asia, that people actually came from the gourds. And depending on how the people exited the gourds, says something about the social ranking of people. So in one mythology, the gourd is pierced twice, once just kind of drilled and once with a hot iron rod. Out of the side that's just drilled come people of lighter complexion. Out of the side that's charred come people with darker complexion. And from this and the way they come out kind of talks about how people are viewed at that time. And these myths change over time. So in some of the older versions, you see more traditional communities of Southeast Asia named. And once you get into more modern eras, you might start to see Americans and French and other people being added into the stories as people from different countries come into Southeast Asia. Another place is in architecture. So here we have the Baipuya Pagoda in Myanmar and also Wat Kutao in Chiang Mai. And besides in architecture, you can also see these shapes in a lot of pottery as well. So if you can use some of your imagination, a bottle gourd, something like this might have inspired this WAP. And maybe something like this would be the inspiration for the Baipuya pagoda. But as you look across Southeast Asia, you can start to see all these similarities to the bottle gourd. So I'm telling you all these historical uses of bottle gourds, but what does this really have to do with plant breeding? Well, when plant breeding is defined, we also think about things about, maximizing positive genetic traits, higher production, resistance to pests, pathogens, and drought. But what does plant breeding really mean? So one thing I'd like everybody to do at the end of this presentation is take out a piece of paper and write down your definition of plant breeding and think about what plant breeding really means to each of you. Because when I read some of these definitions that tend to be more technical, it's not quite how I envision plant breeding. And when I think about plant breeding, I feel like the missing link in some of these definitions is the people. Yes, having resistance to diseases, drought tolerance does help people in a way, but it doesn't really think holistically about how those changes change the systems they go into. So as I've been characterizing the USDA bottle gourd collection and taking some of these coursework in Southeast Asia history, it's really changed my perspective of how I think about plant breeding. So just to give a few examples of things I've started thinking about. So one is thinking about how gene flow and bottlenecks mimics how this crop has been transported to different countries. Can we look at some of these trade routes between countries in Southeast Asia, but also to Africa, to China, to Europe, and see how these crops are moving and maybe see points of genetic diversity or mutations? Another thing with characterizing is some of these uses have phenotypes that we might not first think about. Using a gourd as a resonator. Well, what makes a good resonator? Is that a phenotype that we should be capturing that's important to people? It's not something I probably would have thought of firsthand, but there's all these traits that may be very important to these communities culturally that we're not capturing. If we don't capture them in the germplasm diversity, there's a potential that these traits will no longer exist. And, here thinking again about culture, and, a lot of these can kind of relate into plant breeding. So obviously, we think most about food, but thinking about folk art and celebrations, that's maybe not something we first think about in plant breeding, but these things are really important to different communities. So with that, thank you for listening to my presentation. And do you guys have any questions? Yeah, Travis.
Yes. Do you have any feel for how much of the gourd areas, let's say, are characterized already and how much you're still missing? Did you say some majority still remains to be characterized? We have a good graph on
So, I can only speak to what the USDA has because that's what I've most been working with. There's a lot of phenotypes present, and there is about 174 accessions that are currently available that we used in the trials. And I guess one of the benefits / some of the hard things working sometimes with the USDA germplasm is it's not always uniform. And a lot of it, the accessions are a little bit more like populations than they are like a breeding line or a cultivar. So I think because of that, we're capturing a lot more of the diversity. Unfortunately, there's still, we only had 1 or 2 accessions from Southeast Asia. I think we still capture some of that diversity we see in Southeast Asia, but I don't know if maybe there's different mutations that are causing maybe the same trait that we're not capturing, looking at the genetic diversity standpoint. One thing I'm trying to do is start looking at germplasm collections in other countries and think about maybe capturing the genetic diversity and working with them. But I guess that's kind of my concern is there's definitely certain countries that are like very represented and ones that have very low representation. And I mean, if you don't have it, you don't know what's really there and if it's really something different or if it's really the same as what is it from these other countries. Any other questions? Ed?
In this is, I guess you didn't talk very much about using it for fishing.
For fishing?
Yeah, as a bob for INS. When I was taking archaeology many years ago, this is what the argument was. They were used as early science in fishing.
Mm-hmm.
That's still a whole, this is not a whole water.
Yeah. For when I wrote the paper related to this talk for my class, I didn't really get anything for fishing as far as Southeast Asia. I have heard about using them in bobbers, and I think people still do quite often. And I think part of that might just be like the sources I was able to access. And I wish I could like look at more archives, but I only speak a little bit of Thai. But I think if I was able to kind of go into those archives and look more, I would be able to find a much larger diversity of their uses as well. Ooh, this is something I think about a lot. So most of the collection comes from Africa, I believe, Zimbabwe, and then from India. And then there's kind of a smattering of things from the Middle East, a couple from Southeast Asia, and a few from Europe. There are a couple collections. I believe the World Vegetable Center has quite a few accessions from Southeast Asia. So there's a potential that that area is maybe a little bit more covered, but just not in USDA context. Other countries, I'm not so sure of. And there is use of bottle gourds on every single continent except for maybe Antarctica, but not into the degree as I think it's used in Africa and through Asia. I'm very interested in doing exploration, especially to see what farmers are still currently using. Unfortunately, from my experience trying to submit plant exploration proposals, it's quite difficult. And one of the really big barriers is they want you to have experience going on plant exploration, but there's very few people left who have gone these explorations and would like to continue doing them. So I would say that's kind of the big barrier right now for doing some more collections. But I think just doing some more work, figuring out what maybe smaller collection exists may not need to actually do a plant exploration.
Great.
Any
Thank
other
you for your
questions?
answer.
Yeah.
Just wondering if your seminar is fantastic. Reminded me a little bit of like last week. These I really love this very diverse type of talks. So I'm wondering for your dissertation, what do you see other chapters of the vision overall scope?
Oh, that's a good question. So I'm still kind of debating how I want to split my dissertation. So part of the work I'm doing, I want to create a formal crop characterization guide for bottle gourds because there are no formal crop descriptors. And as I've been going through previous studies that look at bottle gourds, it's really hard to tell whether this shape is really the same shape or they're talking about something slightly different. So I'm hoping that that will be a chapter. For the USDA collection, we took leaf tissue samples for dark seek. So hoping to look at allele frequency and hopefully run a GWAS as well. The third chapter is a little bit up in the air. There's potential. I kind of would like to talk about some of the cultural aspects of this because I think it's a really important thing. I've been trying to do a lot of research looking at the care and fair principles and maybe trying to write something talking about this project and those principles and thinking about characterizing these more traditional vegetable crops.
Okay, thank you for the introduction, Charlie. Once again, I am Savanah Dale. I am from Mike Bohr and Joyce Van X Labs. And today I'm going to be telling telling you about some work that I've been doing adapting a practical haplotype graph for imputation in sweet corn. So sort of to give you some background on this work, low vitamin intake is a modern food challenge in the USA. And by low vitamin intake, I don't mean clinical levels of low vitamin intake, but sub-recommended levels of vitamin intake. So a few compounds that we're really interested in sweet corn are provitamin A carotenoids, antioxidant carotenoids, and vitamin E. So these compounds, when you don't get enough of these in your diet, what can happen is you can have increased risks of conditions such as blindness, maternal mortality, or increased risk of cardiovascular disease. And sweet corn offers a great opportunity for addressing some of these issues because sweet corn is a very popularly consumed vegetable in the US. As of 2019, it was the 6th most consumed vegetable according to the USDA. And it also has natural variation for these fresh kernel nutrient quality traits, including vitamin E and carotenoids. So previous work that has been done in the Gore Lab has used a sweet corn inbred line association panel that was created by Bill Tracy and Matt Baseggio. And this was used to identify inbred lines that had desirable alleles for genes that we know contribute to these fresh kernel nutritional quality traits. So after we selected these inbred lines with desirable alleles, what we did was created F1 biparental populations. And from here, for the rest of our breeding pipeline, we are selfing these to the F5 generation, at which point we will conduct yield and quality testing and generate high vitamin inbred lines. However, as every plant breeder knows, we are constrained by time and space. So what we want to do to speed up this pipeline is integrate marker-based selection in early generations. So keep in mind, these will be highly heterozygous individuals that we want to perform the selection in, ideally our F2 generation. And this necessitates a method for efficient genotype imputation. So this is where the practical haplotype graph, which was created in the Buckler lab, comes into play. This can be used to accurately impute genotypes from skim sequence data. So just to give you a very, very brief overview of a PhG, I wish I had more time to go into this, but a PhG is, when we say graph, this is what we mean by the graph. It is a graph of different consensus sequences that have been created from a starting panel, in this case, a pangenome of different maize accessions for each region in the genome. And the number of regions is variable depending on who creates the PhG and also what the end user wants. So the way this works is we would start out with our reference genome here. So for maize, this would be something like B73. And then a low coverage whole genome sequence genome. This would be the accession that we want to impute. And the reference, we would align this to the reference. This reference genome is the same reference genome that was used to create the PhG that I showed you. So what happens is we split our low coverage or skim sequencing data into the same regions that the PhG includes. And then we can actually compare each region where we have known data. So this would be the orange block, the green block, and the blue block. We can compare these to our PhG, say this is most similar to the orange block in the PhG itself. The green block is most similar to the green block, the green consensus sequence in the PhD, and the blue block is the same as this blue consensus sequence. But we have this area of missing data here in our skim sequencing. And what we want to do is use the PhD to predict or impute what this missing data is. So the PhD uses this graph of consensus sequences and uses a hidden Markov model and a Viterbi algorithm to actually predict the most likely path through these consensus sequences to then predict, okay, this is the most likely haplotype that we see in this region where we're missing information. So then we can fill in the blanks, so to speak. But the use of a PhD raises a lot of questions. And I think the biggest one is, does the PhD that you're using need to include your population founders? So that's the big question for me. I've been working with the Buckler Lab using their maize pangenome PhD, but this doesn't include any of my sweet corn population founders. So is there enough genetic representation in it to perform imputation in my populations? So what I'm starting out with is I have 9 sweet corn inbred founders, which I have 72x WGS data for. My 4 biparental populations of about 200 individuals each, which I have 0.8x WGS data for. And then I have this control IA453 line from the Resende lab, which I have 22x WGS data for. And then like I said, I'm using an existing maize pangenome PhD from the Buckler lab. And then I want to impute genotypes for my populations at the end. But I need to test if this PhD has enough genetic representation to accurately impute genotypes in my F2 progeny. So to start with, I am using this method, this pipeline that I have here, to input my 9 inbred sweet corn founders. So I'm not doing anything with my F2s at this point, just to clarify, as well as my control line. And this is very high coverage whole genome sequencing data. I aligned it to the maize pangenome. So this is my reference genome. And then I'm using the existing maize pangenome PhD to predict haplotype paths or impute genotypes for my founders and my control line here. And this IA453 is my control line because it is one of the 3 sweet corn accessions that were used to create the pangenome PhD. So ideally, it would perform better than any of my lines. So once I have these predicted haplotype paths, I'm converting them into pseudoreferences, which is a term that Ed very wonderfully gave to me. So thank you, Ed, if you're still here. And then aligning these pseudoreferences, which is basically just a FASTA of my predicted genomes for each of these lines, to the actual sequence data. And then I'm calling variants from those alignments. So what this is doing is allowing me to look at the differences between the predicted sequence and the actual sequence. So if the imputation works perfectly, I would expect there to be no difference between my actual data and my imputed data. But what we can more realistically expect is that in my pseudo-reference, the imputed founder sequence, there will be gaps or miscalls in this sequence. So what I did, I ran the pipeline that I've shared with you so far. And then I looked at the number of SNPs that were called. So these are the number of variants between the actual and imputed sequence, and got a lot of information, but we're still working on how to really interpret this. What does this mean? But I'd like to draw your attention specifically to, I subsectioned out this graph and converted it to show SNP percentages. So on this y-axis, I have SNP rate or percentage of SNPs of all base pairs that were imputed, how many are imputed incorrectly. So this is ranging from a little under 0.0006% up to 0.0017% incorrect imputations. Oh, I would also like to point out this IA453 again is my control line, and this is what I expected to perform the best. So when we're interpreting these, we want them to all be 0. Everything should be 0 in this graph. If it was a perfect imputation, that's not what we're seeing. And I'm also seeing my control line did not perform as well as I would have expected it to. And I hypothesized that this could be due to the representation of sweet corn that is in the pangenome PhD. Particularly, so here's my control line, which again is one of the sweet corn lines that were used to create the pangenome PhD, along with P39 and IL-14H. What I would have expected is that FA56A, which is my founder that performed the best out of everything, would be more closely related to one of the sweet corn lines that was used to create the pangenome PhD. But that's not what I'm seeing based on this phylogenetic tree that was created by Vincent Calantonio, formerly of the Resende lab. So thank you, Vincent. So that is my hypothesis. So what I'm doing now, I've showed you this pipeline and just a few of my results from my work so far. And I am actually adding that information from, I use the variance, so those differences or miscalled SNPs between my imputed and my actual sequences, added that information back into the PhD. So now we have an updated pangenome PhD with information from my sweet corn founders. And I'm performing the same steps again. And what I would expect to see is, this is what we saw before, where I have these pseudo references, where I have gaps and mistakes. I would expect to still see some mistakes in the new imputation that I'm generating, but fewer because the PhD database has been updated with new information. Unfortunately, I have not completed this at this stage, so you guys will have to stay tuned for later. But the eventual goal is to then use the existing PhD, an updated version of the PhD, as well as another very commonly used method of imputation, and compare these and their imputation accuracy in my F2 progeny sequences, my SCEM sequence data. So with that, I would like to thank the Gore lab, the Resende lab, the Buckler lab, especially for all of their help with this work, and all of my funding sources. And I will take any questions at this time. Yeah, Travis.
Are you planning for your error rates to look at what their distribution is across the genome? Like, are you making errors in gene regions versus coding regions, stuff like that?
Yeah, that's a really good question. So the question was, am I looking at distributions of error rates, whether they're occurring in genic regions or intergenic regions? What regions of the genome are we seeing the most error and imputation in? And the answer is yes, I will be doing that, but I have not done that so far. So that's a really good question, because if the errors are occurring in mostly intergenic regions, it might not matter. And this is a very good point for PhD users, is how much accuracy do you actually need to be able to get the data that you need for your project, right? So that's a question that I want to answer with my work. But also, especially in my case, we know if we have some particular loci that we're interested in, if we have good imputation in those loci, then we're probably okay to move forward. So thank you. Good question. Yes, Ed.
Yes. Again, I really like that, but yeah, I wonder, at the chromosomal scale, it'd be very interesting to see whether or not there's variant errors across. Okay, because I think there's a lot of hidden Markov models can absolutely weird regions of the genome with bad mapping reads probably go off the rails, and it's a good evidence cluster function as opposed to a rare alleles.
Yeah. Yeah, thank you, Ed, which again is supporting what Travis said, that looking at where these mistakes are occurring is really important. And also if these are occurring in rare alleles, which this is also a really good point because part of this is if you are really looking at rare alleles and you want to have a low error rate in your rare alleles. But in particular, that's important when you're looking at very highly heterozygous lines like I am as well.
This has been a production of Cornell University, on the web at cornell.edu.